are other forms of, okay, other forms of cell death that contribute here as well. So understanding kind of the two camps of cell death, we have apoptosis. So that's, this very typical, this is the kind of process that happens over, millions, if not billions of our cells every single day. It's totally, necessary. It's actually anti-inflammatory. So it actually sends out anti-inflammatory signals to the cells, including, resolvin mediators and other lipid mediators as well. And that requires a phagocytosis. On the other side of it, of course, is necrosis. And, this idea that the cells are exploding, releasing all of their intracellular contents. And those damage-associated molecular patterns, or DAMPs, are really associated with signaling to the immune system that there's something wrong. So this necrotic cell death is definitely a pro-inflammatory immune trigger. And then there's something that you can kind of almost consider to be right in the middle, and that's programmed necrosis. So the end result is the same kind of necrosis that everyone's always known and loved, but it's programmed. So every step of the way, there's a signal, there's a checkpoint, there are inhibitors, just like there are throughout the apoptotic process. So walking through then, what are the differences between, or how is necroptosis triggered and differently than apoptosis? It starts usually with a TNF receptor engagement. This main kinase, RIP kinase 1, is actually usually there to restrict cell death. So an inflammatory cell doesn't usually want to die. It wants to stay alive and fight, damage or infection. So normally RIP1 will induce NF-kappa B. Or TNF will continue to signal if there's engagement of the receptor complex here, and the cell will undergo apoptosis. If, however, caspases are disengaged, whether they're exhausted, they're inactivated, there's inhibitors, RIP1 now becomes a scaffold for a phosphorylation and phosphorylated. That recruits this other RIP kinase 3, that recruits MLKL, which gets phosphorylated, and then MLKL goes to the membrane, ruptures the cell membrane, and all of this stuff comes out with a DAMP. So this is really the key steps in the necroptotic pathway. So just like apoptosis, it's programmed, it's regulated, but it does not use caspase. It is caspase independent. So we, of course, started to wonder if necrotic cell death contributes to inflammation and atherosclerosis. And I won't go over a lot of this. This has been published a while ago, but we and others, of course, have been looking at this. And we find that if we reduce the activation of necroptosis using a small molecule inhibitor, so in this case, necrostatin 1, we can reduce sort of the necrotic core, we can reduce plaque size in mice with established atherosclerosis. If you genetically delete RIP3, same thing, you get much less advanced atherosclerosis compared to in the presence of RIP3. We haven't published this work yet. It's coming out hopefully soon, or we're in the process of it. Who knows how long that's going to take, inhibiting RIP1. So that upstream mediator, RIP1, also reduces atherosclerosis. And then in unpublished work, separately than this, we also show that this is involved in obesity. So then, but the thing about necroptosis is this marker, phosphorylated MLKL. And that, again, that's that committed step that ruptures the membranes. And we had noticed that in human plaques, you could see in advanced plaques, these little brown dots here, which are a little hard to see, but they were really only present in the advanced atheromas. They weren't present at all really in anything that was considered intimal thickening in humans. So we then wondered if we deleted MLKL, we knocked it down, would that reduce necrotic core formation and atherosclerosis, just like it does with RIP3 or RIP1? So this is the work that Adil has been working on for a while, who's going to tell you a little bit more at the end. So he took ApoE knockout mice, he treated them, he fed them a Western diet, typical Western diet, and treated them with Ionis Pharmaceuticals, gave us these great antisense oligonucleotides, or ASOs. So either control ASO or ASO targeting MLKL, and we just looked at lesions over a period of time. And like we suspected, we do see reduction in necrotic core in the mice. So because that lysis of the membranes isn't happening, the actual, the lesion necrotic core is lower. So that was kind of, nice to see and a little bit of what we expected. However, plaque area was not actually decreased. In fact, it always trended towards being increased. It didn't make a lot of sense to us, but this was, no matter how many times we did this or what we analyzed, we always came to this conclusion. So somehow, despite a reduction in necrotic core, we were getting increases in plaque area, or at least not decreases. So we checked, and that the markers of cell death in the plaque, so using tunnel, for example, it did definitely indicate to us that there was little to no cell death going on in the plaque. So very, very little when you inhibit the MLKL with the ASO. So again, that's what we were expecting if you prevent that rupture of the cell. However, there was a lot more oil red O. So unexpectedly, despite the fact that MLKL is gone and the cells aren't rupturing, there's quite a bit more lipid area or oil red O positive area in these mice. So we started to investigate this a little bit. This was actually really surprising to us. So what Adil noticed is when he actually took macrophages and he loaded them up. Sorry, I think actually, I don't remember if these are loaded or unloaded. Maybe Adil, you can let me know. But when he stained with things like Bodepi or Philippin, he actually found that knocking out MLKL actually reduced or increased the amount of lipid that these cells actually had. So they became a lot more foamy. So these, this kind of correlated in vitro, and these are peritoneal macrophages with what we had been seeing in vivo. So he went on a little bit to say, to figure out, what might be happening. And with, anything regarding cholesterol trafficking, you think, okay, maybe it's efflux or uptake, or, maybe ABCA1 is affected or CD36. And the short answer is none of those are affected. So cholesterol efflux doesn't actually change at all with MLKL knockout. That's up here, up on the top left here. And then this is the expression of ABCA1 and CD36 doesn't change. So it doesn't seem to be that the uptake or efflux machinery is altered in the presence of MLKL. So it's not a mechanism regarding uptake or efflux. However, it turns out MLKL actually has other functions that have been described that have nothing to do, or at least we don't know what the regulation of these functions are with respect to its role in cell death. So others have shown that MLKL actually binds phosphatidylinositol phosphatates in the cell membrane to mediate its oligomerization at the membrane. But it also actually binds these structures within the escort and RABs during vesicle secretion. So it's not just that MLKL goes to the membrane and punches holes, but it actually is interacting with escort machinery, the multivesicular bodies, and secretion of vesicles. Now, a lot of this had been done mostly in the context of cell death. So, this was kind of maybe how a cell executed death or avoided death. And nobody really knew if this had anything to do with, any other function of MLKL in any other sense. So Adil then looked to find whether or not any of these markers of the endosome or the multivesicular bodies or any of the other, trafficking machinery were found together with MLKL. And he did indeed find that MLKL co-localizes with markers like RAB7 and CHIMP4B in the multivesicular body and the late endosome. And when you lose MLKL, you see quite an accumulation of CHIMP4B. So those multivesicular bodies actually seem to be increased when you lose MLKL. PIP3 again is that phosphatidylinositol phosphates in the membrane, and there's more of them again when you don't have MLKL. So we think kind of what's happening is that there's almost like a backup of these vesicles, and that the actual lipids cannot traffic out of the multivesicular body endosomal pathway to then get to the ER and stimulate efflux and get to where they need to go. And that's kind of why they get stuck. And that could be, potentially why we're seeing this, excessive oil red O in the plaque itself. So we think that, these are the mechanisms that are really contributing. So it means really that MLKL is this cell death protein that is actually constantly trying to, maneuver the cell between cell survival and cell death. So under normal steady state circumstances, we think that MLKL sort of enables the proper trafficking of the multivesicular body to the lysosome, and then of course all of the downstream pathways that occur. And if you don't have MLKL, we see that really all of this is just backed up, in particular at the late endosomal multivesicular body stage. So it kind of points that this necrosome or necrotic cell death is really at kind of a junction with cell survival and trafficking and lipids. So now Adil's going to tell you a little bit now about the next step of this project and how we also observe some other crazy things that MLKL is doing. And I'll let him take it away from here.
Okay, thanks, Katie. So I can just hit share screen, right?
I think so.
Nope.
Nope.
It says host disable participant screen sharing.
Uh-oh. Okay. Let's do this. How do I do that, Ebru? Do you know?
It's done. It's done.
It's done? Okay, so you should be able to do it now, Adele.
I think you have to stop sharing
Oh,
this.
I have to stop sharing. Okay.
Yeah.
Okay, hold on a moment. Hold on a moment.
Difficulties.
We practice this, but not with, you know, not with everything. Hang on a second. I don't know why it's not letting me do this.
I think you have to go all the way to the top.
There we
and
go.
Click
There we go.
start.
Yeah.
Okay.
Done.
And now I can start.
Switch
Perfect. Can you guys see the presentation?
Perfect.
Okay, so
Now you have to go to presentation mode.
is that working?
Yeah.
Awesome. All right, so thanks for giving me the opportunity to share this bit of my research. It's actually one of my interests, which is hematopoiesis. So what we noticed about MLKL expression, looking at like looking online for whole transcriptome data, is that MLKL's expression is among the highest in inflammatory macrophages. So similar to what Katie just mentioned, we noticed that there was a expression of MLKL, and we've described that role in the inflammatory macrophages, in addition to necroptosis, that being trafficking of the cholesterol. But MLKL is also very highly expressed in hematopoietic stem and progenitor cells. So just like a really quick overview of hematopoiesis, this typically occurs in the bone marrow, where hematopoietic stem cells are found in a quiescent environment, and they encounter growth factors and cytokines, as well as they bind to niche cells, and this promotes their quiescence and prevents their differentiation, as well as prevents their egress into circulation. But when there's a systemic cue, or a change in the growth factors and cytokine profile in the bone marrow, as well as a disruption of the interactions between the HSCs and the niche cells, this primes HSCs to egress and differentiate to the immune cells that we find in circulation, such as the lymphocytes and myeloid cells. But during disease, hematopoiesis can actually occur outside of the bone marrow, and this is termed extramedullary hematopoiesis. So for instance, during atherosclerosis, hematopoiesis can be shifted from the bone marrow to the spleen, where there's premature egress of the HSCs out of the bone marrow, and they seed in the spleen, where hematopoiesis occurs. But what's interesting about extramedullary hematopoiesis is that actually HSC differentiation is more skewed towards this myeloid population and away from the lymphoid population. So during atherosclerosis, the spleen will provide, but while the bone marrow microenvironment's been really well defined, and we know a lot about the interactions between the HSCs and the niche cells, as well as have defined the niche cells and the growth factors and cytokines that exist in that environment, the factors regulating hematopoiesis in the spleen are a little less well defined. Nevertheless, during atherosclerosis, splenic hematopoiesis provides an excess reservoir of monocytes, which then travel to the arterial wall to further promote lesion progression. And what we noticed in our MLKL knockdown model, so again, this is what Katie described. So it's 16 weeks of Western diet feeding in the ApoE knockout mice that administer the ASO. We found that MLKL ASO treated mice saw an almost threefold increase in their spleen weight. And so there's really two major compartments of the spleen. The first being the white pulp, which is outlined here in green, are these purplish areas. And so the white pulp consists of lymphocytes, and it's responsible for active immune processes. And then there's the red pulp, which is this red area, which is the area of active hematopoiesis. And so in the red pulp, you can find niche cells, as well as hematopoietic stem cells and myeloid cells. So we wanted to ask whether MLKL knockdown altered the composition of the spleen. But first, we wanted to see where MLKL was expressed in the spleen. And you can see here, MLKL is in red. It's actually quite nicely restricted to the splenic red pulp. And this is an ApoE mouse after chow diet. However, after we feed the mouse a Western diet, you can see that while MLKL is still in the red pulp, its expression level dramatically decreases, suggesting that MLKL has an endogenous role on splenic hematopoiesis. But going back to the pulps of the spleen, you see that in the control ASO, this is what the spleen looks like. So there's large areas of white pulp, and the white pulp look very multilobular in appearance. However, when you knock down MLKL, in addition to the spleen getting bigger, there's also reductions in white pulp area seen here, and expansion of the red pulp. And when you quantify this, there's an almost threefold increase in the red to white pulp ratio after MLKL knockdown in the spleen, consistent with the increase in spleen weight that we previously observed, together suggesting that MLKL is restricted to the red pulp of the spleen, and that decreased MLKL expression either endogenously by feeding a Western diet or by pharmacological means by knocking down MLKL with the MLKL ASO, this decrease in MLKL expression is associated with the expansion of the splenic red pulp. So this led us to our hypothesis that MLKL may therefore be an endogenous repressor of splenic hematopoiesis, and this may additionally restrict atherosclerosis. So again, this is just the general hematopoietic scheme. I'm not going to show the data, but we also did this in the bone marrow, and we observed no changes in any of the progenitor cell populations. So beginning with the HSCs, when we knocked down MLKL, we saw there was a significant increase in the proportion of HSCs in the spleen after MLKL knockdown. However, compared to this increase in HSCs, we saw a decrease in both lymphocyte progenitors as well as mature lymphocyte populations in the spleen. However, consistent with the increase in HSCs, but opposite to what we see in the lymphocytes, myeloid progenitors such as a common lymphocyte, megakaryocyte, erythrocyte, and granulocyte monocyte progenitors are all increased along with mature myeloid populations. And you see this, the skewing away from the lymphocyte population and towards the myeloid population is a pretty good indication that we actually are seeing extramedullary hematopoietic phenotype. So this suggested to us that MLKL represses hematopoiesis in the spleen, but not the bone marrow. Now, this just might arise from the numbers of cells. So we wanted to ask whether MLKL knockdown intrinsically affected HSC, the ability of HSCs to differentiate towards cells of the myeloid lineage. So for this, we obtained splenocytes from the controller ASO treated ApoE knockout mice and seeded them in this methylcellulose media containing IL-3 and GM-CSF. And this is to push their differentiation and produce myeloid forming colonies. And when we scored the colonies, we noticed that there was an increase in the granulocyte, monocyte, and total colonies in the samples obtained from the splenocytes from the MLKL knockdown mice. So you can see here there was a significant increase in all myeloid colonies after MLKL knockdown. But this is whole splenocytes. So this consists of both the hematopoietic stem and progenitor cells, as well as mature myeloid or lymphocytes, as well as other niche cells such as endothelial cells or stromal cells. So to further refine the assay, we then immunomagnetically selected the HSPCs from the splenocytes and performed this assay. And what was really interesting to us was we completely lost the phenotype of increased myeloid production after MLKL knockdown when we selected only for the HSPCs. So this suggests to us that MLKL regulates hematopoiesis in the spleen through a non-HSPC target. So this could be, again, mature immune cells such as macrophages, regulating the splenic microenvironment, or it could be non-immune cells such as endothelial cells or the stromal cells. And so this is really where our studies are going from now. We're trying to further refine these assays and pinpoint where MLKL might be having its effect. So what cell type and what mechanism that might be undergoing. So just as a quick summary, during atherosclerosis, splenic hematopoiesis occurs, which produces monocytes that enter the arterial wall and contribute to plaque formation. But during the loss of MLKL, there's an increase in the splenic hematopoiesis, which in turn provides an excess reservoir of monocytes, which further contribute to plaque formation. So our future studies will be using tissue-specific freeze in order to delineate where MLKL might be having its effects, whether it's in the HSCs, stromal cells, or in other mature immune cells, such as macrophages. And one other piece of data that we didn't show here today was that when we knock down MLKL, we see a systemic decrease in circulating cholesterol levels. So that might be related to the increase in cholesterol content in the plaque that we find after MLKL knockdown, which is related to the results that Katie shared. But in other models of hematopoiesis, excess hematopoiesis, there are reductions in circulating cholesterol that are observed. So to figure out whether this is the effect of enhanced lipid retention in the macrophages versus increased utilization as a result of splenic hematopoiesis, I guess we are performing this assay. We're performing this experiment now, but COVID kind of messed up our timeline a little bit. But we're going to splenectomize the mice while giving them the Western diet and the ASO and see whether that can normalize cholesterol levels. And so, so I'd like to thank, obviously, the entire Rayner lab who have been instrumental in these studies, my collaborators, as well as, Ionis for providing the ASOs and the funding sources. And I guess we'd be happy to take any questions.
Great, thanks, Adele.
So thank you, everyone, especially Adele, for a wonderful presentation. So we're happy to take some questions. If you want to ask a question, please use the participants window and raise your hand. It's a blue button. Perfect. So I'll take it in order. So Cheyenne, so I will unmute you. Thank you. Go ahead, Cheyenne.
Thank you, Katie.
Oh, you're cutting out there, Cheyenne. Oh.
Yeah, thank you, Katie.
Uh-oh.
Oops. Okay.
Nice talk. I was wondering in the MLKL ASO study. Oh, sorry. Can you hear me perfectly now?
Now we can hear you.
No.
Now we can hear you, yeah.
Not anymore.
Still, cannot hear me? Okay.
Yeah.
I was wondering in the ASO, sex differences in the ASO studies.
Are you asking if there were sex differences, I think?
Yes.
Yes, sometimes.
No. I mean, very seldomly. So when we looked at like immune cells, obviously the plaque area was significantly higher in the females versus the males. But in terms of the effect between the ASO and the control, sorry, the MLKL ASO and control ASO, I don't really think that there were.
Thank you.
Wen Dai.
Hey, hello.
Yeah, we can hear you.
Hey, Dr. B. Nice to see you again. Hi, everyone. I'm from Dr. Alaska's lab, and I have a question. Is that this is very interesting topic, Adele, and because we know that the extramedullary hematopoiesis is very important in humans when in the embryo stage, but when for the adult, for the people, and then the bone marrow takes the role. So I was wondering for the mice, how much the extramedullary hematopoiesis contribute to the blood cells, especially for the spleen, and also for the mice work you check. I was wondering the age of the mice you used. Thank you.
Adele, you want to take that one?
Okay, yeah. So thanks for the question. We started the mice at 8 weeks old, and then fed them the Western diet for 16 weeks, so they were about 24 weeks old at the time of sacrifice. We have looked at immune cells, but in circulation, but we haven't been able to track them necessarily between the bone marrow and the spleen. So unfortunately, we don't know the relative contributions of each compartment to contributing to overall immune cells, either in the plaque or the peripheral blood.
Yeah,
Okay.
and I'll just add to that Adele didn't sort of have time to get into this too, but some of what we think about the stromal cells versus the hematopoietic stem and progenitor cells is that there may be an interaction between them in terms of their numbers for sure, or their differentiation, but also whatever they're secreting. So we find a few interesting candidates in the plasma actually that are different. So it may be that there's something happening, with respect to the spleen and the way that it affects stem and progenitors, but or it actually might be related to what they're producing and actually the growth factors and cytokines that they're spitting out. So we have lots of work to do on that end too.
Yeah.
Okay, thanks.
Thank you, Wen. Lynn Hedrick.
So this is actually Klaus. Hi, guys. I have a quick deal. So about 10 years ago, Alan Tall reported in ABC A1G1 double knockout mice this extramedullary hematopoiesis and myeloid skewing. And I was wondering whether you directly compared the phenotype of those spleens and the cell counts. Thank you.
No, I haven't directly compared it, but that's a great suggestion. We definitely note that they're different mechanisms. So, or we suspect, sorry, that they're different mechanisms. So the A1G1 really works directly on the hematopoietic stem cell, but what we're seeing, I believe, is more of an effect on the stromal population. In terms of direct cell counts and looking at the proportions, we haven't done that. I have looked at ABC, A1, and G1 in the spleen, and I believe that the expression levels aren't that different. So yeah, it'd be interesting to go back and make a direct comparison. I'll definitely have to do that.
One thing though that may be similar, and Adil and I, not that we disagree on this, but we go back and forth between this all the time, is more that because in our foam cells, we see that MLKL really well, others have shown this too, it really binds to the PIP3 in the membrane. So it's possible that although the sort of effects maybe may not be in the progenitor cell like was observed with ABCA1 and G1, it may be still through sort of this raft-like, membrane fluidity, potentially signaling through the IL-3 receptor, so those kinds of things could actually be very similar to what observed with ABCA1 and G1. Actually, I thought for sure we would just see like messed up ABCA1, and that was going to be our mechanism. A1 and G1, I thought for sure, and I don't think that's it. But good question.
Thank you. So Prashant Tevkar.
Thanks for the great talk. Thanks for the great talk. So my question is, like during the progression of the disease, what percentage of the monocytes are contributed towards the from the myeloid, from the bone marrow hematopoiesis versus the splenic hematopoiesis? Is there any point where one takes over the other, or is there a balance from where these cells come in?
Yeah, so in general, you mean in atherosclerosis, or in this model, do you think, or are you asking just in general?
No, in general.
Yeah, I don't know, Adele, if you know those kind of numbers off the top of my head. But,
I don't know the numbers, but in certain cases, such as with the GCSF model, so this is commonly used for for cancer therapeutics, especially in the mouse models, when you have an extramedullary hematopoietic, a very extreme model like that, you actually do see exhaustion of the bone marrow. And that's because there is rapid differentiation, rapid egress of the HSCs. And the HSCs that don't readily, differentiate into immune cells in the circulation will seed in peripheral tissues, and those peripheral tissues will then produce the myeloid cells. In our model, we don't really see a lot of exhaustion of the bone marrow in the MLKL knockdown versus the control. So I'm not, I wouldn't suspect that the bone marrow in our model isn't contributing to a lot of the myeloid cells. I think that it is still very functional. In the knockdown versus the control, again, we haven't done those studies, but I think that's something that's really important, and we might have to address with a spleen transplant.
Thank you. So Ada.
Hi, thanks for the great talk. I have a couple questions. The first one is in relation to what Klaus asked, and I was wondering if you actually measured the amount of cholesterol in your progenitor cells.
No, we haven't done that yet. That's definitely something that I'm interested in doing. We have pardon?
We have the samples.
Yeah.
And my second question is about the MLKL ASO treated plaques. So it had been shown by single cell sequencing that the foamy macrophages are not necessarily the inflammatory type. So are your MLKL ASO plaques less inflammatory? And did you check that?
Good question. We didn't look, actually, to be perfectly honest. We didn't do any kind of particular plaque stains. I think in general, none of the cytokines in the serum were different, but that's just the serum, right? So that's not telling us a lot, but no, that's actually a really good suggestion. We could look specifically in the plaque and look for all the, the various kinds of inflammatory markers. Yeah, we should do that.
Thank you, Ada. So last question from Fangli. Fang.
Yeah, can you hear me now?
Yes.
Yeah, I'm just wondering whether splenic hematopoiesis will also cause increased monocyte like numbers in the plasma as well. So
Yeah.
check. Okay.
In general, they do. Like in the normal models of extramedullary hematopoiesis in response to hypercholesterolemia, that's exactly what happens. There's sort of that reservoir in the spleen, and it makes its way into the circulation, and it makes its way into the plaque. But interestingly, when you have this MLKL knockout, this loss of MLKL, the circulation, there's not really much of a difference in the circulation. The spleen goes wild, but the circulating monocytes don't really change. So we don't know, is that because they're not getting out? Is that because they're spitting out a factor? Are they not differentiating? I mean, we don't know. It's really, normally, yes, it would translate to in circulation.
Okay, but you do able to observe increased macrophage area in the plaque?
Most of the macrophage area is, I mean, the plaques are not bigger really. I mean, they're trending towards being bigger. They're just not really significantly bigger, but they have a lot more lipid. So on a kind of per macrophage basis, they've got more lipid content, but not necessarily more macrophages. It seems that they're probably a little tiny bit bigger because they're physically, you know, accumulating a little more lipid, but it doesn't translate into tons more macrophages per se.
Just one more question for Marit. So
Oh, great. Thank you very much. And thank you, Katie, for the great talk, and Adil as well. I was wondering, because when I actually wanted to go back to the comment of Klaus Ley, because he mentioned the extramedullary hematopoiesis in the ABCA1 and ABCG1 knockouts. And we actually found that that was dependent on GCSF and the GCSF secretion by macrophages and dendritic cells. I was wondering, did you look at that in the setting of your MLKL mouse?
Adele?
Yeah, so we've done the gene expression for GCSF, and it is actually increased as well. Unfortunately, we've looked at several different factors, both hematopoietic and non-hematopoietic, so it doesn't lend us to kind of narrowing down on the mechanism, but we have looked at GCSF, GMCSF, MCSF at the gene expression level in the spleen so far, and we are planning to do an ELISA and further characterize whether this is similar to the A1, G1, ApoE, if this is potentially a common beta subunit of the IL-3 GM-CSF receptor dependence.
Yeah, but it was not dependent on the common beta subunit. So the proliferation was dependent on that, but not the stem cell mobilization. So the experiment that we eventually did is that we blocked the GCSF and then we blocked the extramedullary hematopoiesis. It's something that you may want to do.
An experiment that Adil literally finished the day we shut down. Like the day we shut down was the GCSF stimulation in the presence and absence of MLKL, basically, so that we could kind of characterize in a more traditional extramedullary hematopoiesis whether or not this is playing a role. Is this hypercholesterolemia only that's really, you know, because obviously that's where this all came from, was hypercholesterolemia, but we're trying to see, and then, you know, we'll have more data when we get back to the lab, I guess.
Yeah, of course. Thank you.
Thank you.
So that will be great. When you get back to the lab, we can hear the rest of the story.
Yeah, exactly. Yeah.
Happy to share. Thank you.
Thank you so much. I'm going to hand it over to Lale, Ed, and Ada. Thank you, Katie and Adele.
Okay, great. Our next talk will be from Ed Fisher's group. As you know, Ed is one of the pioneers in atherosclerosis research, and today Eda from his group will tell us about their exciting work on the regulation of atherosclerosis resolution by Wnt signaling. And I will ask Ed to introduce Eda.
Yeah, thank you. It's actually, I'm getting a kick out of today because Ada is an outstanding postdoc at NYU. She won the award actually last year as the outstanding postdoc, followed that up with a K99 this year. And we are following the talk from the laboratory of another outstanding postdoc from NYU, Katie Rayner, of course. So Ada, we have just by coincidence today, two great advertisements for the lab, Ada and her talk. And actually I just received the cover picture from JCI Insight, which has a beautiful figure from one of our papers on neutrophil extracellular traps, which we will not get into in Ada's talk. But as you heard from Lale, we're going to give you some surprising information about Wnt signaling. and the resolution of atherosclerosis. So Ada, with your beautiful background that you had of the Empire State Building, let's go to your talk now.
Thanks, Dr. Fisher, for the introduction. And thank you for having me here, giving me the opportunity to present our work. And also, thank you for giving me the excuse to change out of my pajamas. So I'm going to tell you today about this story I've been working on for the past few years. And if you just want to tune out afterwards, I'm just going to give you the take-home message now. And this is basically what we found. So in resolution of atherosclerosis, we found that IL-4 signals through STAT6, and that is required but not sufficient for resolution of the disease. And actually, to get full, robust response, let me just put a pointer here. To get full, robust response to IL-4, there's a need for a second signal. And we found that in resolution of atherosclerosis, this second signal is PGE2, or prostaglandin E2, signaling via STAT3. And this PGE2 is produced in response to Wnt signaling. So now for the data. You all know how atherosclerosis develops. You know, we're in a pandemic, we sit in our homes, we order in burgers, we binge watch Tiger King, and we binge eat ice cream, and we get disease development. And most therapies today aim at stopping the disease when it's already advanced and people already have symptoms. And our lab believes that the best way to resolve this is actually to clear the arteries, clear the inflammation, and regress the disease. And this is a process that in the lab we call resolution of atherosclerosis or regression, and I'm going to use those terms interchangeably. In the lab, in the past 15 to 20 years, we've established a few mouse models for resolution of atherosclerosis. And these have commonalities in the way that we do the experiments. And the mice we take mice with plaques, so hyperlipidemic mice. And after they develop plaques, we use methods to reduce the cholesterol and revert them to normal lipidemia. And importantly, we sometimes we see changes in the size of the plaque, but more important to us is the composition of the plaque. So in regressing plaques, we see fewer macrophages, and the macrophages that are in the plaque are also of a more remodeling flavor and anti-inflammatory flavor. And as you know, this is not as easy in humans that you can just reduce their cholesterol and get disease resolution. But we think that these mouse models are very important so we can find the pathways that are important for resolution and then use them in humans. So already almost 3 years ago, we published this paper in which we used the aortic transplant model to examine STAT6 involvement in the resolution of the disease. So in this model, we take arches from hyperlipidemic mice, so these arches contain plaques, and transplant them into normal lipidemic, either wild type controls or STAT6 knockout mice. And in this model, the arches that are being transplanted, they are sufficient for STAT6, and only the cells that are being newly recruited in the resolution phase are deficient for STAT6. And our data shows that there's resolution of atherosclerosis, so fewer macrophages in the plaques from the wild type recipients, but this is impaired in the STAT6 recipients. And when we look at the flavor of these macrophages, again, the gene expression of these macrophages, the wild type recipients, and this is done by laser capture microdissection or LCM from aortic plaque macrophages, we saw higher expression of pro-resolving genes such as arginase 1 in the wild type recipients, which was again impaired in the STAT6 knockout animals. So from this, we concluded that STAT6 is required in the cells that are being newly recruited to resolving plaques. And this is where my project started. And we wanted to know what are the signals that are upstream of STAT6 that promote this resolution of atherosclerosis. And we started by looking at the classical activators of STAT6, which are the cytokines IL-4 and IL-13. So the first experiment we did, we took IL-4/13 double knockout animals, or wild-type animals. Now these are total body knockouts. And we made them hyperlipidemic and harvested these mice after 20 weeks of hyperlipidemia. One group of each genotype was then reverted to cholesterol reduction regime, and we harvested those mice after the cholesterol reduction phase. Now, first thing we saw, which was very obvious, was that the wild-type plaques were larger and had more macrophages than the IL-4/13 double knockout plaques. And this is in line with other publications in IL-4 knockout animals showing that IL-4 is actually also important in progressing plaques. Nevertheless, the plaque size in the IL-4/13 knockout animals was still substantial, so over 300,000 micrometers. In regards to resolution of disease, we saw a significant reduction in macrophage content of the plaque in wild-type animals, but not in the IL-4/13 double knockout animals. So this means that IL-4/13 are important for resolution of atherosclerosis, and without them, we see impaired regression. Now, our next question was whether the source of the IL-4/13 is from newly recruited cells, because STAT6 was needed in newly recruited cells. So we turned back to our transplant model. And now, again, we had our baseline group that are hyperlipidemic, and we took the arches from these mice and transplanted them into either wild type or IL-4/13 double knockouts. And again, here in this model, the arches contain cells that are sufficient for IL-4/13, but the cells that are being newly recruited to the plaque after the transplant are deficient for IL-4 and 13. And interestingly, and we did not expect this, the resolution was comparable between the wild-type recipients and the IL-4/13 knockout recipients. So the macrophage reduction was similar between the two groups, meaning that the IL-4/13 recipients had the same amount of resolution of the disease as the wild types. So IL-4/13 is not needed in the newly recruited cells, but actually in the cells that already reside in the plaque during the progression. Now, to see if there is more production of IL-4 and 13 in resolving plaques, we again did a regression model in vivo, and we scraped the plaques carefully and measured by ELISA the amount of these cytokines. So as a control to see if this actually works, we looked at MCP1, which we previously showed both by mRNA levels and also protein levels to be decreased in resolving plaques. And again, when we did this ELISA, we saw a clear reduction in MCP1 in resolving plaques. Surprisingly, when we looked at IL-4/13, we saw these data. So this was a surprise for several reasons. One is that IL-13 was not detected at all. This maybe suggests that IL-13 is not important in the microenvironment of the plaque. And when looking at IL-4, we saw two interesting things. One is that its levels are not changed in resolution of atherosclerosis. And the other interesting thing was that its total abundance was around 100 picograms per ml, which is 200-fold less than what we usually use in vitro experiments. So for this reason, we focused on IL-4 going forward, because we didn't detect any IL-13 in the plaques. And whenever we did in vitro experiments, we used this plaque-relevant concentration of IL-4 of 100 picograms per ml. Now, it's also possible that the total abundance of IL-4 is not changing in resolving plaques, but maybe there are more IL-4 producing cells that increase the local concentration of the cytokine. So to examine that, we did another regression model with IL-4 reporter mice. So these mice have GFP expression in each cell that expresses IL-4. And again, we had a baseline group, a hyperlipidemic baseline group, and one group was subjected to cholesterol reduction. And we looked at the GFP expression of those cells in the aortic arch. And again, we did not see any significant change between baseline and resolving plaques. And we did this also in an aortic transplant model. We didn't see any more IL-4 producing cells in resolving plaques. So to summarize what I showed you so far, I showed you that in progressing plaques, there's accumulation of IL-4 and IL-4 producing cells. And then after cholesterol reduction, when monocytes come in and become plaque macrophages, they become pro-resolving plaque macrophages. And that is through activation of STAT6 in these newly recruited cells. But the main question here was how are these monocytes now responding to IL-4, although the abundance of IL-4 does not change? And a couple of years ago, there were two papers from Carla Rothland's group and Judy Allen showing that actually to get a full response to IL-4, you need a second signal. In Carla Rothland's group's paper, they showed that this second signal is apoptotic cells. And in Judy Allen's paper, they showed that this second signal is tissue-specific complement factors. And we were thinking maybe the same is true also for resolution of atherosclerosis, that there's a second signal that is needed. And what does this second signal may be? And we're thinking about Wnt. Why Wnt? So before I joined the lab, the Fisher lab had this paper showing that macrophages from resolving plaques have an increase of the expression of these genes that are all regulated by canonical Wnt signaling. We also went back to a recently published data set from aortic macrophages in progressing and resolving plaques from our lab published last year. And we looked at the cumulative expression of Wnt-related genes. And we saw that in resolving plaque macrophages that there's a higher expression of these Wnt-regulated genes. So again, in an independent model, we verified that plaque macrophages in resolving environments have more Wnt signaling. And this is just an overview of what the Wnt signaling is. It's a molecular on-off switch in which beta-catenin is always being produced, but also always being degraded by this degradation complex. Wnt's are secreted soluble proteins, and when they bind to the receptor frizzled and co-receptor LRP5 and 6, they cause the recruitment of the degradation complex to the membrane, freeing up beta-catenin that can go to the nucleus and serve as a transcriptional co-activator. And this is a particularly interesting signaling pathway in atherosclerosis because it had been shown that there are families with a mutation in the LRP6, the Wnt co-receptor. And these mutations cause a downregulation of the Wnt signaling pathway and also cause early coronary disease. So again, is Wnt actually collaborating or synergizing with IL-4? That was our next question. And to answer that, we looked at bone marrow-derived macrophages and their response to Wnt and IL-4 stimulation. And in our in vitro studies, we used, again, the low concentrations or the plaque-relevant concentrations of IL-4. And we used an activator of the canonical Wnt signaling pathway. And here I'm showing you just the expression of arginase 1 as an example where we have more genes that act similarly. So when we treat cells with IL-4, they increase their expression of arginase 1. But when we co-treat them with Wnt and IL-4, you see this synergistic expression of arginase 1. And this was STAT6 dependent because when we used STAT6 knockout bone marrow-derived macrophages, we didn't see the expression of these pro-resolving genes and did not see the synergy between Wnt and IL-4. So it seems like in vitro there's an enhancement of IL-4 response with Wnt, but is Wnt important in resolution of atherosclerosis? So we did another regression model in which we had, again, baseline hyperlipidemic mice, and 2 groups were then subjected to cholesterol lowering regimes, with 1 group treated with a Wnt inhibitor called LGK974, and the other group treated with a vehicle as control. And this Wnt inhibitor actually inhibits the secretion of Wnt, so the availability of the endogenous Wnt that is being produced. When we looked at macrophage content of the plaque, we see that in control plaques, there is a clear reduction in macrophage in the plaque, but this was impaired in the Wnt inhibitor group. When looking at the flavor of the macrophages that are still in the plaque, we saw that the control plaques contain macrophages that express more M2-like markers and less M1-like markers. And this was impaired in the Wnt inhibitor group. So the Wnt inhibitor group had plaques that were resembling of the baseline animals. So to summarize what I showed you so far, I showed you that in atherosclerosis resolution, there's a need for IL-4 and STAT6 signaling, but this is not sufficient. So it's required, but not sufficient. And there's a need for a second signal. And we think that this second signal is through Wnt. And our next question was, how does Wnt enhance IL-4 responses? So we took a look again at our bone marrow-derived macrophages that were treated with Wnt. And we did RNA sequencing on these cells and saw an increase of the JAK-STAT signaling pathway. And this was interesting to us because we know that STAT6 is another STAT family member, STAT3, sorry, is another STAT family member that can have pro-resolving effects because it's downstream of IL-10. And IL-10 is another pro-resolving cytokine that was shown before to collaborate and synergize with IL-4. And we also repeated these studies. And actually, we also found that IL-4 can synergize with a pro-inflammatory cytokine that is upstream of STAT3, and that is IL-6. So to examine whether STAT3 is involved, we first examined whether Wnt can activate STAT3. And STAT3 is activating through phosphorylation. We treated bone marrow-derived macrophages with Wnt and indeed saw phosphorylation of STAT3 16 hours post-treatment. Interestingly, we did not see this phosphorylation at early time points. So up to 1 hour of Wnt treatment, we do not see this phosphorylation. Then we knocked down STAT3 in bone marrow-derived macrophages. And again, in control cells, we could see this enhancement of IL-4 responses with Wnt and IL-4. But this was abrogated in cells that were knocked down for STAT3. And in vivo, we went back to our Wnt inhibitor study, and we looked at activation of STAT3 in those plaques. And we saw much more STAT3 nuclear localization in the control-treated animals and less in the Wnt inhibitor-treated group. So it seems like Wnt activates STAT3, and this is required for the enhanced response. Now, how is STAT3 doing this? So we hypothesized that Wnt promotes the production and release of a factor that can then bind to a receptor and activate STAT3, because classically STAT3 is activated downstream of cytokine receptors. To examine if that is the case, we did an experiment where we made Wnt conditioned media by treating macrophages with Wnt for 16 hours. And then we treated naive cells with Wnt conditioned media. And you can see here in this Western blot, this is what I told you earlier, that Wnt itself in short time points, so 15, 30, and 60 minutes after exposure to Wnt, there is no phosphorylation of STAT3. However, if we expose the cells for the same amount of times to the conditioned media, to the Wnt conditioned media, now we see a rapid phosphorylation of STAT3. So it seems like this is actually happening where STAT3 promotes the production and secretion of some the Wnt promotes the production and secretion of some factor. So what is this factor? For this, we collaborated with Matt Spite, and we did metabolipidomics of the condition media, of the Wnt condition media, and saw that 16 hours after the treatment, there's an increase in all these metabolites, and most of them were metabolites of the arachidonic acid pathway. Of these metabolites, we're particularly interested in prostaglandin E2 or PGE2 because it had been shown before that it also synergizes with IL-4, and it can also promote the phosphorylation of STAT3 in multiple cell types. So that fit very well with our data, and we wanted to examine if PGE2 is actually produced by Wnt-treated cells and can synergize with IL-4 in our system. So we checked that in 2 different experiments. In the first experiment, we looked at cells treated with Wnt3A again and IL-4, and again saw an enhancement of arginase 1 expression. When we treated the cells with conditioned media and IL-4, we saw similar enhancement of arginase 1 expression. When we blocked Wnt3A itself in the conditioned media, again, we saw the enhancement in the synergy between this conditioned media and IL-4. But this enhancement was completely abrogated if we treated the Wnt conditioned media with a blocking antibody to PGE2. In a reciprocal experiment, we blocked the receptors for PGE2. So here again, in control cells, we can see the enhancement of IL-4 response when treated with Wnt or with Wnt conditioned media. We actually saw the same enhancement if we blocked the EP2 receptor or the EP4 receptor, but this enhancement was abrogated in cells that were inhibited for both the EP2 receptor and the EP4 receptors. And just to loop back to in vivo again, we again took a look at the regression study we did with the Wnt inhibitor. And here we did laser capture microdissections of plaque macrophages and looked at the expression of enzymes that regulate PGE2 levels. And of these enzymes, we saw a reduction in the PGE2 synthase in mice that were treated with a Wnt inhibitor. So to conclude what I showed you here today, I showed you that in regressing plaque macrophages, there is a higher response to Wnt signaling, which enhances the expression of the PGE2 synthase that promotes the production of PGE2 that via EP2/4 receptors signals through STAT3 to enhance the IL-4 STAT6 signaling and get the full IL-4 responses. Our next steps is to understand which cells are producing Wnt, because it had been shown in the past that every cell type in the body can produce Wnt. We actually don't even know where the macrophages are responding to Wnt. It's possible that they're responding in the microenvironment of the plaque, but it's also possible that they're responding in the bone marrow, the circulation, the spleen. We don't really know where. And now we want to also understand why is Wnt signaling lower in progressing plaques and higher in regressing plaques. So, of course, I want to thank all the people that did the work. Especially thank you for Dr. Fisher for your support and mentorship. Thank you for the Fisher lab members who helped with this project, the previous lab members and the current lab members. Thank you for our collaborators, our bioinformatician collaborators, Matt Spite and Brian for their help with the conditioned media experiment, Kiara, Don, and Nick for some great discussions, Rich Loxley and Tom Wynn for mice. And I'm happy to answer any questions now. And if people have questions later, you can Twitter me or email me. Thank you.
Hey, great talk, Anna. So if you have any questions, yep, I see that Henry, please go for it.
Hi, yeah, Ida, this is great, and congrats again to your grant. Yeah, I guess my question is that, is there any way to measure the extracellular level of PGE2 in the plaque? Because you would imagine that if the cells begin to produce and secrete more PGE2, the cellular level of the PGE2 itself should be lower, but the extracellular will be higher. That's relatively easy to model in a cell culture dish, but in the plaque, I would imagine it's really challenging to confirm that.
Yeah, I agree. It's very challenging to confirm. We are doing some lipidomic studies in the lab to see the difference in the different lipids in plaques, but I think it's also very hard because what we see is in macrophages, right? And it's also possible that maybe other cells produce some other things, or that the total amount of PGE2 in the plaque is not increased, but it's the local production by the macrophages. So it's even more complicated than what you stated, but we definitely are interested in doing that.
Okay, great. And then we have another question from Bushra. Yep, Bushra, go for it.
It was a great presentation. Thank you so much, Ada. My question is, you always added, as far as I see, exogenous wind to the media or exogenous wind injection. So do you plan to do endogenous reporter assays to measure the activity of the wind in the macrophages, like with the AL4 knockout and the wild type mice? So it could be really interesting to also see the actually endogenous wind activity is changed with the top-lesh assays, for example.
Yeah, I totally agree. I think it's a great point. We are doing some experiments with reporter mice. I just have to make the point that, yes, in our in vitro studies, we do add exogenous Wnt, but in our in vivo model, actually the inhibitor that we used is an inhibitor of the endogenous Wnt because it inhibits the secretion and the availability of the Wnt that are being produced. So we were actually really excited to see such an effect with this inhibitor.
Okay, thank you.
Okay, great. And then next one is Ben Porset. Ben, go for it.
Yes, hello. Thank you for this very nice talk. We have shown actually a few years ago with Ed Fisher and Ines Pineda-Tora that LXR does play a role in the regulation of these processes. And well, obviously, when you activate LXR, you altogether with IL-4, you do see the same effect on arginase 1 expression. And it has been shown that LXR and Wnt interplay together, and there is a crosstalk between both pathways. So I was wondering if you've looked in your experiment whether LXR is also activated, or if Wnt regulate the phosphorylation, because obviously LXR phosphorylation also play a role in these processes, and maybe that could be a first part of an answer and of your new questions.
Yeah, I think it's a great point. We didn't look at LXR activation at all, but it's something that we should definitely do going forward.
Okay, next question is Jesse Williams.
Hey, Ada. I think I was a little confused about the origin of where IL-4 is coming from and which cell you think it's actually signaling through. Since you have all that single cell seq data, did you guys define which populations producing it locally?
Yeah, that's challenging, I think, with the single cell data. But with our reporter mice, we actually see that it's both the T cells and eosinophils. It depends on our mouse model.
Do you have good imaging data that complements that flow data you showed, just to see kind of where those cells are in relation to the macrophages?
We have those sections and we can do that, but I don't remember having imaging data. But that would be, yeah, that would be an interesting thing to look at.
Okay, and last question is Ebro. Ebro, please go for it.
Thank you for a great talk, Ada, and congratulations on your award again. I have a question. So mine is a little bit organellocentric. Since you see a lot of impact on the prostaglandins as a common denominator, have you checked whether your peroxisomes are stressed or intact or affected?
We haven't checked, but we should.
I just thought that would be an interesting point.
Yes.
Okay.
I have a question too, Ada, actually. I can't raise my hand, I think, as a
Go
host.
for it. Yeah, Katie, go for it.
So that was awesome. I love seeing the progression, no pun intended, of the work. I'm wondering, because the Wnt LRP kind of pathway, in addition to beta-catenin, if I'm not mistaken, there's also sort of a cholesterol mobilization, kind of lysosomal mTOR connection as well. I'm wondering if you've looked at how that kind of signaling might be impacting your regression phenotype, in addition to potentially the PGE secretion.
We haven't specifically looked at mTOR. The good and the bad thing, I guess, with Wnt is that so many things are happening, and it's activating so many things. So I'm gonna have a long career investigating this.
Okay, great. So I think that's the end of it. We thank the speakers, amazing talks, and we'll see you next week again, same time. Thanks, everyone.
Thanks, everyone.
Bye.
Nice to see everybody.